Hello friends, today through this video we'll discuss the nuclear reprogramming of induced pluripotent stem cells. In previous video we have already seen the characteristic and its types of the stem cells, and in this video the induced pluripotent stem cells can be defined as the artificial stem cells which produce from an adult cell that have regained the capacity to differentiate into any types of the cell. There is a term nuclear reprogramming, and the term nuclear reprogramming is used to describe either functional or the molecular changes to cells undergoing fate changes. The Shinya Yamaka, Japanese physician and the researcher and colleagues, by using the mouse cell, it was first described by them. So several proteins have been identified that are capable of inducing pluripotency or enhancing non-pluripotent adult cells on the transcription factor octamer 4, the SOX2 and the BOX2, which maintain the stem cells in a primitive stage. Other proteins like KLF4, NANOG and the GLIS1. There are three major approaches through which the nuclear reprogramming is achieved: the nuclear transfer, cell fusion and the transcription factor transduction. So we'll see each in one by one. So first is nuclear transfer. When a nucleus from a differentiated somatic cells, intestinal cells, is transplanted into the diploid enucleated oocyte, the nuclear reprogramming is initiated. Through a figure we can clearly see there is a oocyte in which the nucleus is removed. Then it is called as the enucleated oocyte, and the somatic cells, which is from the patient cells, the nucleus is removed and it is fused with the oocyte, and then the blastocyst is formed and it in the individual. So somatic cell nuclear transfer is done. The first successful cloned mammal was Dolly ship made by the nuclear transfer. So there is a somatic cell nuclear transfer, which is very important in the purpose of examination view, and this experiment showed that the developmental stage of a nucleus from adult somatic cells can be reprogrammed upon its transfer into an unfertilized oocyte. Next one is the cell fusion. In this, the embryonic stem cells and the somatic cells is fused to form the foran, and then there is a two type of division like no cell division and DNA replication to leads to the heterokaryon, while the cell division occurs and DNA replication occurs to form a same type of species which is haploid in number, and differentiated species which is aneuploid hybrid. So in cell fusion, the two distinct cell types are combined to form a single entity, and the resultant fused cells can be heterokaryon or the hybrid. What is heterokaryon? It is short-lived and do not divide. As a result, they are multinucleated, and if the fused cells proliferate, they will become hybrid, and on division, the nuclei fuse to become tetraploid. So next one is this transcription factors transduction, and in this case, the somatic cells, this approach can be used to form induced pluripotent stem cell, which have similar property to embryonic stem cells and can be generated from the almost any cell type in the body through the introduction of the figure of the transcription factor transduction are as follows. In this, the four types of genes are used. The octamer 4 genes play a vital role in deciding the fate of the inner mass and embryonic stem cells, while the sex-determining reason Y plays an essential role in maintaining the self-renewal potential in embryonic stem cells. KLF4 and the MYC has an important role in cellular proliferation and the metabolism. So there are three methods by which the induced pluripotent stem cells are produced. The induced pluripotent cells have numerous biomedical applications in basic research in drug screening, toxicological studies, disease modeling, and the cell therapy. So this is about the nuclear reprogramming of induced pluripotent stem cells. Thank you and keep watching.